Hello everyone and welcome to my channel EG Life Science. Here in this video we are talking about some questions answers with explanation from a subject techniques in biochemistry. So subscribe my channel EG Life Science. If you like my video, share it to your friends. So let's start. The first question: paper chromatography is especially useful for the detection and separation of. So the correct answer for this question is option 4, that is amino acids. By using paper chromatography, we can separate and we can detect the amino acid in a mixture. The paper chromatography is a technique used for a separation of compounds based on a differential solubility in a stationary phase and a mobile phase. In the amino acid separation by a paper chromatography, the solvent travels across the paper and it carries the amino acid with it. Paper chromatography is an analytical technique for separating and identifying the mixtures that contains colored compounds, especially the amino acid and pigments. It is a method of separating a dissolved chemical substance by the different migration rates across the sheets of paper. And in a paper chromatography, we calculate the RF value. The RF stands for retardation factor, and this retardation factor is the ratio of the two distances. One is a solute's distance and another one is a solvent's distance, and this is a ratio for RF value calculation. Another types of chromatography are there, that is a column chromatography, and this column chromatography is one of the most common method for protein purification. Ionization chromatography, it is most commonly used for protein separation, and in ionization chromatography a positively charged matrix is called as an anion exchanger because it binds negatively charged ions or a molecule in a solution. Then the gel filtration chromatography is used when an aqueous solution is utilized to transport the DNA containing sample through a columns. HPLC is one of the such kind of chromatography, high performance liquid chromatography, and this is used for a separation of DNA fragments up to 20 kilobase pairs with high resolution. TLC is a thin layer chromatography. It is a versatile technique for a separation of liquids in a sample or a mixture. So the correct answer for the question is option 4, that is amino acid. The next question, here we can look for the image of column chromatography, paper chromatography, and a thin layer chromatography. The liquid chromatography separate a liquid sample with a liquid solvent, that is in mobile phase, and column composed of a solid beds, that is a stationary phase. In a gas chromatography, the gas chromatography separates vaporized samples with a carrier gas, that is a mobile phase, and a column composed of liquid or a solid beds, that is a stationary phase. Then the paper chromatography, this chromatography separates the dried liquid samples with a liquid solvent, that is a mobile phase, and a paper strip, that is a stationary phase. Thin layer chromatography separates the dried liquid sample with a liquid solvent, that is a mobile phase, and a glass plate covered with a thin layer of alumini or a silica gel, that is a stationary phase. Then the next question, it is a most commonly used filter paper for a paper chromatography, and the answer for this is Whatman number 1 paper. It is a most commonly used filter paper for a paper chromatography. The next question, Whatman number 1 filter paper is mainly composed of? The answer for it is cellulose. The method of electrophoresis is developed by, or it is given by a scientist, that is Thales, on the basis of electric charge or movement of the particle in electric charge. the scientist Thales invented a electrophoresis technique. Combination of electrophoresis and a chromatography is called as, answer for it is a ionophoresis technique. Then next question, ultracentrifugation method was developed by, four options are there. Out of the four options, the first option that is Thales, he found a electrophoresis technique. The second option that is Swedberg, and the correct answer for this question is Swedberg. The Theodor Swedberg, he is an inventor of ultracentrifugation. Ultracentrifugation is a technique separation of on the basis of a centrifugal force created by a very rapid rotation up to the 50,000 revolutions per minute or more than it. Different molecules depending on their masses will settle at the different speeds under these conditions. The centrifugation uses a low speed for a separation, whereas ultracentrifugation uses a high speed for a separation. The Theodor Swedberg is an inventor for the ultracentrifugation, and the Swedberg constant for ribosomes 30 years, 60 years or 70 years like that. This S stand for a Swedberg coefficient, and this coefficient it is on the basis of the settlement rate of the ribosomes, on the basis of their settlement rate. The Arne Thales, option A, he gave us a electrophoresis technique. He was the Swedish biochemist, and he won a Nobel Prize in chemistry in 1948 for his research on electrophoresis. So the correct answer for this question, that is Swedberg. The 3D structure of protein can be determined by, 3D structure of a protein can be determined by NMR as well as X-ray crystallography. So the answer for this question is option C, that is both A and B. We can make a use of NMR and X-ray crystallography for determination of a 3D structure of a protein. The 3D structure of a protein is determined by a technique such as X-ray crystallography and a nuclear magnetic resonance. The protein tertiary structure is a three-dimensional shape of the protein. The tertiary structure will have a single polypeptide chain, that is a backbone with one or more protein. A secondary structure, and they have the amino acid side chains. They may interact and bound in a number of ways, and the interaction and bonds of side chains within a particular protein determines the tertiary structure of the protein. The NMR and X-ray crystallography are the answer for this question. So C is a correct answer. The spectrophotometer. The spectrophotometer measures the number of photons emitted to estimate the intensity of light spectra absorbed or transmitted by a sample, and this provides the information on the amount of compound in a sample. The NMR, it is a spectroscopic technique to observe a local magnetic field around atomic nuclei. The sample is placed in a magnetic field, and the NMR signals is produced by a excitation of nuclei sample with radio waves into a nuclear magnetic resonance, which is detected with sensitive radio receivers. X-ray crystallography, it is a experimental science to determine the atomic and molecular structure of crystals, in which the crystalline structure causes a beam of incident X-ray to diffract into many specific directions. By measuring the angles and intensities of these diffracted beams, a crystallographer can produce a three-dimensional picture of the protein. Then the next question, disulfide bonds are formed between. The disulfide bonds are formed between a cysteine residue that are close together. Answer for this question is option A, cysteine residue that are close to each other or close together. Formation of disulfide bond between cysteine residues occur during a folding of many proteins that enter the secondary pathways or a secondary structure. Formation of disulfide bond in a protein occurs between a cysteine side chains via the oxidation of a thiol group. The cytosol is an unfavorable environment for a disulfide formation because it contains one enzyme, that is a NADH dependent reducing enzyme, and because of this such kind of disulfide formation does not occur in a cytosol of the organism. The disulfide bonds are a central structural element which stabilize the 3D structure. They are mostly found in secondary proteins and extracellular domains of membrane proteins. So the correct answer for this question is option A, that is a cysteine residue. In between cysteine residue there is the formation of disulfide bonds. Which of the polysaccharide is often called as animal starch? In an animal there is a glycogen. We know that in a plant the storage or storage material of a carbohydrate is a starch, but in case of animal that is a glycogen. So it is called as an animal starch. Which of the following country is a major producer of agar agar? The answer for this question is the option D, that is Japan. The agar agar powder is produced from the marine algae, that is Gelidium, and this agar agar powder, the major country for production of agar agar powder is Japan, and in Japan it is known by one name, that is canteen. And this agar agar powder, it has no flavor, no odor or no color, and it is a solidifying agent for a preparation of a culture media. Carbohydrate with a free aldehyde or a ketone group known as. The answer of this question is option A, that is reducing sugar. The carbohydrate with a free aldehyde and a ketone groups known as a reducing sugar. A reducing sugar is a sugar that has a free aldehyde or a ketone group that can act as a reducing agent, whereas a non-reducing sugar does not have a free aldehyde or a ketone group, so it cannot act as a reducing agent. Here are some examples of reducing sugars. Monosaccharides like glucose, fructose and all other monosaccharides are the reducing sugars. The maltose and lactose, these are the disaccharides, these are also reducing sugars, whereas the sucrose, raffinose and stachyose, these are the non-reducing sugars. Here in this image, there are the differences in between reducing sugar and non-reducing sugar. The reducing sugars are carbohydrate that can act as a reducing agent due to the presence of free aldehyde or ketone groups, whereas non-reducing sugars are carbohydrate that cannot act as a reducing agent due to the absence of free aldehyde or a free ketone group. The reducing sugars are a good reducing agent. Non-reducing sugars are not reducing agent, have a free aldehyde or ketone group, don't have a free aldehyde or a ketone group, give a positive reaction toward the Fehling test, gives a negative reaction toward the Fehling test, includes all the monosaccharides and some disaccharides, includes some disaccharides and all polysaccharides. This is the main difference in a reducing sugar and a non-reducing sugar. Here once again, which of the following is a reducing sugar? The answer of it is a glucose. The sucrose is non-reducing, glycogen is a non-reducing. More than half of the carbohydrate ingested by human is a starch. Mixture of glucose and fructose is often called as a invert sugar. Invert sugar is an edible mixture of glucose and fructose, which are both simple sugars. Invert sugar is sweeter than a normal sugar, 125% more than a normal sugar, or it is called as a invert sugar. When the two monosaccharides bind with each other, they form a disaccharide. Here are some examples of the disaccharides. The beta maltose, it is a disaccharide and it is made up of the two monosaccharides, that is alpha D glucose and the beta D glucose. And in between these two sugars or these monosaccharide, there is a one linkage, that is alpha 1,4 glycosidic linkage. So the maltose is a mixture of two monosaccharides, that is glucose and glucose, which are joined with alpha 1,4 glycosidic bonds. Whereas a lactose or a beta lactose, it is also a disaccharide. It is made up of alpha D galactose and beta D glucose. The linkage is a beta 1,4 linkage. The sucrose, it is also a disaccharide. It is made up of alpha glucose and beta fructose. Linkage is a alpha 1 beta 2 linkage. Next question, polysaccharide found in a fungi and arthropods is. The answer of it is a chitin. The chitin is a modified polysaccharide that contains a nitrogen, or it is a amide derivative of a glucose. It is synthesized from units of N-acetyl D-glucosamine, and these units forms a covalent beta 1,4 linkage similar to that of the linkage in between cellulose. It is one of the most important biopolymer in nature. It is mainly produced by the fungi because it is present in a cell of fungi. Also, the exoskeleton of the arthropods and nematodes. In insects, the function of a chitin for a scaffold material or as a scaffold material supporting to the cuticles of the epidermis and a trachea. So the correct answer for this question is option A, that is chitin. The starch, it is not found in a fungi, it is found in plants. The xylan, we know that the xylan is a major component of the plant hemicellulose. After a cellulose, it is a most abundant renewable polysaccharide in nature. Xylan and cellulose are a predominant hemicellulose polysaccharide found in a cell wall of land plants, and xylan is a polysaccharide made up of a beta 1,4 linked xylose sugars. The xylose sugar is a pentose sugar, and it has a branching with alpha arabinofuranose. The dextrin is a group of low molecular weight carbohydrate produced by a hydrolysis of starch and a glycogen. Dextrins are a mixture of a polymer of D-glucose units linked by a alpha 1,4 and alpha 1,6 glycosidic bonds. The pectin, these are a structural acidic heteropolysaccharide contained in a primary middle lamella and middle lamella of cell wall of terrestrial plants. Its main component is a galactouronic acid, a sugar acid derived from a galactose. The glycogen, that is a stored form of a glucose that's made up of many connected glucose molecules. When the body needs a quick boost of energy or when the body isn't getting a glucose from the food, glycogen is broken down to release a glucose into a bloodstream to be used as a fuel for a cells. These are some things related with the carbohydrate, and for this question the answer is a chitin. Then the next question, sucrose is a disaccharide made up of. The sucrose is a disaccharide which is made up of glucose and fructose. Here the sucrose is made up of glucose and fructose. The glycosidic link, that is alpha 1 beta 2 linkage in between glucose and fructose. The positively charged basic amino acids are. The answer for this question is option A, that is lysine and arginine. These are the positively charged basic amino acids, and these are a major amino acids in histone proteins which are required for a chromatin formation in a eukaryotic organism. Among the 20 amino acids, 5 have a side chains which can be charged at a pH 7. 2 are negative charged, that is aspartic acid. The symbol for aspartic acid is a capital D, and the glutamic acid, that is a capital E. These two are amino acids that have acidic side chains. The remaining 3, that is having a positive charged side chains. For example, lysine, the symbol for lysine is a capital K, arginine capital R, and histidine capital H. Means lysine, arginine and histidine, these are the 3 amino acids with a positive charged side chains, whereas the aspartic acid and glutamic acid, these are the amino acids with a negative charge. The here question is asked positively charged amino acids, so the answer is lysine and arginine, option A. If the question is a negatively charged amino acid, then answer will be the glutamic acid and aspartic acid. The aromatic amino acids includes. The answer for this question is option A, phenylalanine, tyrosine and tryptophan. These are the aromatic amino acids. The aromatic amino acids absorb ultraviolet light at a wavelength 250 nanometer and produce the fluorescence. Example of it, phenylalanine, tyrosine and tryptophan. The another option, that is asparagine, it is represented as ASN, or a symbol for it, asparagine capital N. It is a polar aliphatic acid, non-essential in a human, because non-essential means the body can synthesize it. The codon for the asparagine are AAU and AAC. The glutamine, the most abundant in a body, made in muscles. Role of glutamine is for it removes excess of ammonia. It removes a waste. Role in immune system, needed for a normal brain function, and it is represented by a capital Q. It is a neutral polar amino acid. The correct answer for this question is option A, that is phenylalanine, tyrosine and tryptophan. These are the aromatic amino acids. Then sulfur containing amino acids are cysteine and methionine. These two are sulfur containing amino acids, and these are useful for maintaining a secondary structure by formation of disulfide bonds. The simplest amino acid is, the answer of this question is a glycine. The amino acid that has a single hydrogen atom as its side chain are called as a simple amino acid. Glycine is a simple amino acid, and it is represented by a symbol GLY. The codon for glycine are GGU, GGC, GGA, GGG. Glycine is integral to a formation of a alpha helix in a secondary protein structure. It is most abundant in a collagen and it is a inhibitory neurotransmitter. So the correct answer for this question is option A, that is option C, that is a glycine. So thank you for watching my video. If you like my video, subscribe my channel Easy Life Science, share my video to your friends and press the bell icon so you will get the notifications of my videos.